Okay, a bunch of different ways that antibodies can work. The first one, opsonization. Um, that makes a pathogen more recognizable, so your phagocytes can ingest and destroy, eat up, digest anything that's invading your body. Also, it's called neutralization. Prevents viruses from docking. So if they can't get on the surface and inject, get through the membrane, they can infect. Then we have neutralization. Um, they bind to toxins and prevent toxins from affecting cells. So optimization, neutralization, neutralization of toxins. Remember, there's the toxins. You have the cells or the virus themselves. You also have their toxins that can cause injury to your cells. And then what's called activation of this chemical called complement, which those are proteins that make holes. The fancy way of saying that would be perforations. And when you have the holes, water and ions can enter, and what that makes happen is you have lysing of the cell, and that cell can't do anything anymore. It's dead. And agglutination, that makes everything stick together. The pathogens are sticking together. They can't go and infect your cells. They're just stuck to each other. Prevents pathogen from entering the cell. Entering the cell. Entering the cell. And then these large chunks of thick clumps of pathogens then have to get filtered by your lymphatic system, which is kind of a parallel system to your circulatory system. In blood, it's half cells, half plasma, and the lymph takes all the watery substance, the plasma leaks out from the capillaries, and then filters it and gradually returns it to your blood system. And that's why you can feel your lymph glands swell up when you get sick. They can be painful. You can feel them here, sometimes in your armpits. Doctor will check that out. There's a nice little graphics to kind of show you this. This is agglutination. Agglutination. Sticking there, opsonization. Opsonization. Coming around, phagocyte coming around that bacteria, killing it. This is an inflammation. You have some leakiness, so these macrophages can get in there and go after the offending virus or bacteria. And here we have a graphic of neutralization. And we have cytotoxicity. But check those diagrams out. They're a nice way to get a good graphic of it. All right, memory cells. That's what makes you immune. So you can be immune if you have specific antibodies, or mainly memory B cells that can make those antibodies. Okay. You need to be, in order to do that, you need to be challenged either naturally by the thousands and thousands, perhaps millions of pathogens you see every single day your body is exposed to. And don't worry about vaccinations. If there's 2 or 3 antigens in there, that's nothing compared to what you see on a daily basis. And eventually you can create those memory B cells from being challenged like that. You see when your body having those, if your body has those memory B cells, you'll be ready to go. Ready to go. That's ready right there. You see that thing again. So check it out. You have the primary immune response. You get a little bit of a bump in your white blood cells, T cells, but that's you have a weakened form. Say this is from a vaccination. And say you get the secondary exposure, boom, your body has a much larger response and it's faster. Instead of taking all this time to peak, boom, look at that. And then in that same amount of time, you get a lot more white blood cells and antibodies going after whatever is giving you a problem. And usually a vaccine you'll get by injection, although there's some oral vaccinations they'll give to kids, although typically the injection are more effective, at least so far. And you'll either get a weakened or attenuated live virus or a bunch of dead viral parts that your body will be able to respond to. And those, of course, all those dead parts not belonging to you, you know those as antigens. So antigens, oh yeah. So you're getting exposed, but you're not going to get sick. And then the idea is once if you get the actual dose of the virus, your memory B cells be ready to make antibodies quickly and in large number. Remember, clonal selection. That's what this is. Which is kind of a fancy way of saying a whole bunch of mitosis. And so what is happening here? Is color going to work? No. So what's happening here? This is a white blood cell. And it's doing what's called amoeboid. I'm trying to do an M in cursive. Oh man. That's an O. Amoeboid motion. So it's sending out these pseudopods grabbing onto this bacteria and then pulling it in where it can eat it. Okay, so this would be white blood cell or phagocyte. Oh, I'm having trouble with my colors. So eventually it's eating this in. It's kind of the cleanup crew. Clean up bacteria. These are rod-shaped bacteria. Fancy official name is rod-shaped. So right here, species, syphilis, polio, HIV. Zoonotic is a pathogen that can cross species barrier. Here we got bubonic plague, and right here we got West Nile. Very exotic sounding. We get a lot of times this coming close to wild animals, and this is also, remember, HIV. That's how that got started. Humans interacting with wild animals. And then right here is livestock. Organized wild animals or organized domesticated animals. So right here you can see big devastation. This was the San Francisco earthquake. I think it was 1903. And you'll see massive damage, but mostly it was the fires that was the problem. And why are we talking about this earthquake when we're talking about diseases? Because what happens when people lose all their houses, they can't live there anymore. They have to live in camps. And camps become unsanitary. And that can create more possibilities of disease transmission. And that's why here we had hundreds of people die of bubonic plague because they were living near rats that were transmitting that to them. And this is a picture of the SARS epidemic. I believe that was 2006. And you'll see they just had to go through and basically slaughter all these chickens because they were unsure which ones had SARS virus. So they just went through and slaughtered this. This also happened in the 90s, mid-90s, with mad cow disease in England. They just had to go kill all the beef cattle, which was, pretty devastating. Just think about big, huge pits with animals that and were buried. So, that's one of the consequences of, our system of producing, any kind of meat product. We have to remember that could always happen. So, histamine, you guys with allergies, you know about this, make you itchy, sneezy, and they make capillaries leaky, so that plasma leaves the capillary, and that allows the white blood cells in there to go out and fight infections. And phagocytes help to go out and destroy these invaders. Something called mast cells in your connective tissue, like your tendons and ligaments. Help to go also to release those histamine. And that's what makes you feel itchy. So you get cut. Look at the little bacteria right there. And those chemical signals, what is on the bacteria your body recognizes as non-self, starts with an A. You know it. Antigens. So these are the antigens. Look, attracting these white blood cells, aka phagocytes, into that area. Boom. Eating up. See? Eating up all these bacteria, digesting them. What do you notice happens right here around that surface? That's swelling. That's from all these extra white blood cells. Most people, we swell up, we want to ice it, right? That's kind of a bad idea, though. The swelling is there for a reason. It's going to take longer to heal if you ice it. But we're always trying to fight our body's response, so people love to ice things.